We look at the individual proteins and see if they can activate themselves. And also, we expect that, if we see this pathway in one animal, and then we look to a cousin, a distant cousin, it may not have had the same set of mutations. So in different images, you might see the framework of the pathway there, but the individual components might be a little bit different. So those are all predictions that you make from that evolutionary model of how it arrives. Now, go to the next slide. There's our blood clotting pathway, and it fits all the predictions. First of all, in this diagram, you see the little rectangles. That refers to the kind of protein it is. All the proteins that are on those little shaded rectangles happen to be serine proteases. That's just a particular kind of enzyme. It's an enzyme that cuts other proteins. And you'll notice they're all little rectangles, except for the initial step and the right-hand step. So where do these come from? They arose by serial duplication in evolutionary history. Next slide. These are other pieces of evidence. They have gone in and they have done interesting things with mice. We can abuse mice in horrible ways. Genetics has progressed so far. And one of the things you can do, for instance, you can take a mouse and say, well, I want to make a knockout. I want to destroy one of the steps of the blood clotting pathway. And you can do it. It's fairly easy. So, for instance, they even made mouse mutants that lack cyanogen. That's that end step. And according to Mike Behe, that ought to die, right? Saying, if you don't have this, it doesn't function. It turns out they don't die. They actually look pretty healthy. In fact, they're surprisingly healthy. One of the surprising results of this was when they knocked out that protein, the mice grow bigger, stronger, plumper, seem generally healthier, and they don't bleed to death. Amazing. They do have one significant impairment. They can't have children. The females all bleed to death as soon as they get pregnant. About shortly after they get pregnant. So this is one reason why they've still got fibrinogen, obviously. It's got use points. But the total function of the pathway is destroyed. And even the loss of the ability to have children, as you'll see in a moment, isn't an obstacle to the evolution of this particular pathway. When they looked at other lineages, cetaceans lack factor 12, those proteins. Pufferfish lack several of them, factor 11, factor 12, precalcrin. And they all, this is naturally, they lack these things, and they can all clot their blood just fine. And they can also have children, obviously. In Urochordates, these little primitive sea squirts, ancient chordates, there's no elements in the cascade. That entire stuff in boxes in between, it isn't there. When you go a little farther afield and you look into chynoderms, they have none of the cascade either. All they have is fibrinogen on the wall. They just have the end step present in their blood pathways. So what's going on there? Well, first of all, it looks like you can have fully functional clotting systems missing pieces of the story. And actually, when they start piecing together the ancestry of these things, it looks like it makes sense. It's a nice mix of hierarchy in what pieces you have and what pieces you don't have. Some organisms completely lack this, and might be, well, how can they survive? Well, that's what coevolution is all about. So I think that's the next slide. Remember I told you that one answer for IC is that we can build a scaffold, and that elaborate series of steps I just described is building a scaffold. The other is coevolution. In the ancestral state, half a billion years ago, we're all descended from something that would look like a urochordate, a little tadpole-like thing swimming in the ocean. In the ancestral state, they had a very primitive, simple clotting cascade, and that's good enough. Because if you ever get to see a sea squirt, I know we're in North Dakota, not likely, but if you ever get to the coast, look on some pilots sometimes, you'll see the little blobby things, and these are your distant cousins. They're very peculiar looking things. They don't need to bleed much. They don't have hearts that are like ours, that are pumping blood at high pressure through their circulatory system. And so if you cut them, they just kind of ooze a little bit. So pathway is good enough. They have a low-pressure vascular system. And what happened in evolution is, as we got more and more complicated, we became larger organisms. As we went from things like those urochordates to fish, to amphibians, to reptiles, to mammals, what you have at the same time is increasing blood pressure. That our vascular system has become more and more elaborate. And as they became more and more elaborate, they needed better blood clotting. So what was happening over the course of evolution is that you're increasing the complexity of the vascular system, and along with that, you're increasing the complexity of the blood clotting cascade. So that's coevolution. Next slide. The other thing is acceptation. And that's where things evolve for one purpose and acquire a different purpose in evolution. And the example I'll throw out here is the flagellum. This is another favorite of the intelligent design creationists. Flagellum is a kind of whip-like motor that you find in bacteria. And it's a beautiful little thing, very complicated. It's got all these pieces. This is what they're talking about when they say, oh, well, they have rotors inside them. They have little outboard motors. This is a rotor and an outboard motor. The way this works is this thing, this network of proteins here, is spinning around, and it's attached to that whip up there, and that makes the whip flail around and act like a propeller to drive it through the water. How could that have evolved? Look at that. Oh, wow, look at all those pieces. They all have to be there for it to work. Well, they don't all have to be there for it to work, especially if you think about other functions that it could have. So the next slide. When you go looking into bacteria, what you discover is that pieces of this flagella are scattered all over. And the artwork is flagella. For instance, there's something called the ATP synthetase system, which is a proton pump. And this is a set of proteins that the bacteria uses to make energy. What happens is protons move through this across a gradient, and they force it to move, and they also catalyze a chemical reaction that produces ATP, which is, any biology students here know, is one of the fuels of these cells. So it's a little rotor system that moves. It does not have a whip-like thing on it. It just spins around as it's busy making ATP. There's also something called the type 3 secretion system, which has the function of export. And pieces of this look just like the flagellum, only it's not acting as a whip. What it is, it's got a protrusion, and this is one of the nasty evil things bacteria do, is they make toxins in their cells, and they pump them out through this thing to poison other creatures in the environment. And finally, we have a flagellum, which has a little locomotion, but it contains these pieces in it. Next slide. And these can be put together in a nice hypothetical series. That, let's see, up there, the third one over, there's a picture of the ATP synthetase. And you can see what it is. It's a little module that sits in the membrane that spins around to generate energy. And when you look at something like a flagellum, you can find in its core, there is the F1 ATP synthetase. So all these pieces have been co-opted and acquired new functions. But they all evolved for other things. So telling me that one of the early steps in the evolution of this thing would make a really lousy outboard motor doesn't mean anything, because it may have been really good at something else. Next slide. Okay. So those are the intelligent design creations. This is what I mean by their posers. They're putting up these ideas that really don't discredit evolution. I want to say a few words about young Earth creationists. Like I said, they're the majority. There's a lot of them out there. What can we say about young Earth creationists? Well, go to the next slide. I recently made a visit to the Creation Museum in Kentucky. You've heard of this, right? It's amazing. Tons of money thrown into this thing. It's very elaborate. It's a huge building. And what it's supposed to do is supposed to be a museum for the creation side of the argument. When you go inside and know what you discover is that it is not a museum. It's more like a Disney ride. You are shuttled through a series of exhibits that you are supposed to take a moral lesson from. They're supposed to convince you that creationism is true and evolution is false. And this is one of the early exhibits, and it's just a perfect example of what they're doing. They have a predetermined set of events